By the end of the second week of development, the bilaminar embryonic disc, consisting of the hypoblast and epiblast, has formed. Throughout the third week of development, this bilaminar disc differentiates to establish three primary germ layers, in a process known as gastrulation. Around 15 days after fertilization, a thickened structure forms along the midline in the epiblast near the caudal end of the bilaminar embryonic disc. This is called the primitive streak. At this stage, the formation of the primitive streak defines the major body axes of the embryo, including the cranial end towards the head and caudal ends towards the tail, as well as the left and right sides of the embryo. At the cranial end of the embryonic disc, the primitive streak expands to create a primitive node which contains a circular depression, known as a primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak, forming a primitive groove. Once formed, cells of the epiblast migrate inwards towards the streak, detach from the epiblast, and slip beneath it into the interior of the embryo. This process is known as invagination. The first cells to invaginate through the primitive groove invade the hypoblast and displace its cells. The hypoblast cells are eventually completely replaced by a new proximal cell layer, which is referred to as the definitive endoderm. By day 16, the majority of the hypoblast has been replaced. The remaining cells of the epiblast are now referred to as the ectoderm and forms the most exterior distal layer. Some of the invaginated epiblast cells remain in the space between the ectoderm and newly formed definitive endoderm. These cells form a germ layer known as the mesoderm. Once the formation of the definitive endoderm and mesoderm are complete, epiblast cells no longer migrate towards the primitive streak. Throughout gastrulation, the ectoderm continues to form from the cranial to the caudal end of the embryo, establishing three distinct primary germ layers throughout the whole embryonic disc. The gastrulation process is finally complete.
By day 13, the blastocyst is firmly implanted in the endometrium. We'll now make sagittal cuts through each constituent part, one after another. The outermost is the syncytio trophoblast, which secretes the beta-HCG we detect in pregnancy testing. Next is the cytotrophoblast, followed by extraembryonic mesoderm, inside of which is a space known as the chorionic cavity. Sitting in this cavity and also surrounded by extraembryonic mesoderm is the developing embryo, the parts of which we'll discuss next. The epiblast sits atop the hypoblast, and together these are known as the bilaminar disc. Cells of the hypoblast extend out to form this space called the yolk sac. By this stage, the epiblast has developed inside it the amniotic cavity. We'll remove the outer elements in order to take a closer look at the bilaminar disc. At the cranial end of the hypoblast lies the procaudal plate, an area in which columnar cells are present. It serves as a marker of the embryo's cranial aspect. The end point of gastrulation is the formation of a trilaminar disc made up of ectoderm, mesoderm, and endoderm. The cells that make up these layers were all once of the epiblast. And this transition is achieved as follows. Cells of the epiblast migrate inward, downward, and then differentiate. From either side, this continuous passage begins like symmetrical waterfalls. To be clear, it is the moving cells which produce this form, as shown here. Macroscopically, this makes a visible groove in the surface of the epiblast known as the primitive streak. Its presence marks the beginning of gastrulation. At the cranial end of the streak, cells are migrating thickly, forming this circular depression known as the primitive node. As the embryo grows, more cells migrate and the primitive streak elongates. Cells which differentiate into endoderm displace cells of the hyperblast and eventually replace it entirely. Cells which remain in the upper layer differentiate into ectoderm. And cells which differentiate into mesoderm force their way between these two layers. Around day 16, cells migrating through the area known as the primitive node begin to form a hollow rod which curves toward the cranial end of the embryo. It extends toward the procaudal plate and is known as the notocaudal process. We take a coronal cut now. We see the hollow rod, and if we move through it now, we should also see its connection to the primitive node. As the notocaudal process grows longer, the embryo continues to grow in all dimensions. Around day 18, the notocaudal process fuses with the endoderm. From this to this. It's now known as the notocaudal plate. Being continuous with the primitive node, this now allows free passage between the amniotic cavity and the yolk sac. Let's take a look through to solidify that, starting at the primitive node. We're looking down through what is known as the neuroenteric canal, and the space we can see into is the yolk sac. It's theorized that this brief communication between amniotic cavity and yolk sac allows pressure equilibration between the two cavities of the early embryo. Once that has been achieved, the two edges of the notocaudal plate come toward each other and eventually fuse. They become a solid rod, the notochord. Noto is Greek for back, and the notochord acts as the structural backbone of the developing embryo. By this stage, intraembryonic mesoderm fills almost all the space between the ecto and endoderm. Apart from the space the notochord occupies, it is absent also at the oropharyngeal membrane, located where the procaudal plate once was, and the cloacal membrane. These are structural elements of the embryo that currently exist as ecto and endoderm tightly adhered. And that concludes the process of gastrulation, from bilaminar to trilaminar embryonic disc. Hit subscribe for more videos simplifying this complex topic. Thanks for watching, and we'll see you next time.